So if we have a look quickly, I just want to show you one example of how you could be asked this. Okay, they give you a little case study and they give you a scenario. All right, they say a man was stabbed with a knife. The victim managed to scratch the attacker before he ran away. Skin tissue found underneath the victim's fingernail was analyzed for any DNA, and this will be known as the DNA from the crime scene. Okay, the pictures are coming just on the next page, so I'll show you in a moment. And then they tell us that there were 7 suspects, and they had DNA samples from 7 suspects, and they labeled them A, B, C, D, E, F, and G. Now it says, which of the suspects was the attacker, and how do you know? Right, so here is our page. Okay, here, this set of patterns, okay, this set of bars is DNA from the crime scene. Remember, this would have come from the fingernails under the person who was attacked, and it would have been a DNA sample from the attacker. So now what we are able to do is we have to look at all of these 7 samples. Okay, one of the easiest ways in which you can do this is to take a piece of paper or to take something like a ruler, okay, and try and find a correlation between this sample, which we know is the attacker's, and we know for sure because that was under the person's fingernails, okay, so he scratched the attacker. And then you correlate and you look for similarities in the patterns between these suspects that we have. So if we have a look at the bottom, okay, there is a nice line, okay, if you're looking really carefully. Immediately what we can see is that there are not lines at the bottom of samples D, E, F, and G. Okay, but that's not really a good place to stop. Work your way up, okay, and look at the patterns that are here. So if we look at A, okay, A, we can see a thick band over there. We can see 2 thick bands over there, which is kind of in contrast to what we have over here. There, we can also see a big difference. Okay, so we can see a nice thick pattern there, whereas with the sample from the person who had attacked the victim, we can't see a correlation there. Okay, and as we move up, we can see pronounced differences between the two. Okay, so what you would do is you would take each of the suspects and you would compare it. And as we go along, you would see that B is also not going to be a good match. There is a section there which does not correlate with that section there. Those thick bands over there are also not correlating with that section over there. And the point is you would do this until you have gone through all of them, okay? And what we would see is that if we look at suspect C, that band at the bottom correlates. That thick section over there correlates. That thick section over there correlates. Then it becomes lighter and we see consistency there. Then we have a very thick section over there, and the same is true over there. Those patterns seem to be overlapping. So it would be safe to say suspect C is going to be the attacker. How do we know? That was the next part of the question you had to answer. We are able to say conclusively that C is the attacker because the bands or the patterns made when we took a sample of DNA from this suspect match those to the sample of DNA that we had from the attacker. Okay, so you'll be asked either by using photographs like this or by using hand-drawn pictures, which we looked at earlier. Okay, so these are the types of questions that you can expect. Okay, on to another section which is linked to this and very important, is the section of genetic engineering. Okay, genetic engineering is done in a bit more detail in matric, but it's actually not new work. Okay, if we think of genetic engineering and we think back to our grade 11 syllabus, and we think of term 1, and we think of viruses and bacteria, okay, we actually, in that section of work, as well as in another section of work, learned that we can trick bacteria, okay, and I use that word lightly, okay, it's still a very scientific process. which falls under the issue of genetic, or the heading of genetic engineering. But we can trick bacteria into producing, can you remember? Insulin. Okay, so insulin we know is a hormone that is crucially important in maintaining and stabilizing things like glucose levels. Okay, insulin actually changes glucose into glycogen. Okay, so if you've eaten and you have a high glucose level in your blood, we know that that is not a good thing to have for very long. Okay, so insulin is released from the pancreas, okay, from specifically the beta cells in the pancreas, okay, and it converts, it promotes the conversion of glucose into glycogen, and insulin also promotes the uptake of glycogen into your liver and muscle cells so that it can be stored till later. So genetic engineering, okay, is something that we have looked at already. Again, something that is quite important is that we have to know what genetic engineering involves. So genetic engineering is manipulating the genetic composition of an organism, whether it's in this case like bacteria, or whether it is plants, or anything that we have looked at, or that you look at. It's the manipulating of the genetic composition of an organism, which often results in something beneficial specifically for humans. Okay, so there's normally a benefit to humans. Okay, and genetic engineering is a man-made or a man-driven process. Okay, it's something that scientists do, it's something that as people and as humans we are able to do, okay, and we are able to control this process to get some benefit, okay, to us. So just going back to the idea of insulin, I'd like to just show you quickly how this could work, okay. What you do is you take the code for insulin from a pancreatic cell, okay, you isolate that gene, and then what you do is you remove a thing called a plasmid, okay, from bacterial DNA. It looks kind of like this, okay, just in terms of the drawing. It's a fairly simplified version of what happens. But you remove the gene for insulin from a human gene, okay, from a human sample. Using restriction enzymes, okay, you actually remove a section, okay, there they show you nicely. You remove a section of the plasmid, and you insert whatever it is you want this bacteria to code for into its plasmid, okay, which is a ring of DNA. So now the ring is complete, okay, you actually then are able to control this. The bacteria doesn't really know, it doesn't care really what it's coding for, okay, it's just going to carry on coding for it, and it's going to divide and divide and divide, and all the time what you are doing is you are making it code for the gene that you have inserted into its DNA code. Okay, so in that way we are able to manipulate it. We are able to force the bacteria, or allow rather would be a better word, we are able to steer this process and allow the bacteria to make lots of insulin. We're able to harvest it, we're able to take it out, and we are able to bottle it. And this is something that obviously is biotechnologically driven as well. It is linked to genetic engineering because you have actually altered, you have manipulated the DNA of one organism, okay, and you've used DNA from another organism, and by the end of this process we've actually been able to make insulin. Okay, and for people who have diabetes, this is obviously crucial, it's life-saving. Okay, people who are not able to get insulin, or if they have a faulty pancreas and they're not making enough insulin, run the risk of organ failure, they can go into a coma, and ultimately people will die if they don't have one way of getting insulin in. So this is something that really is very useful. There are very low impacts in terms of human involvement here. We don't have to worry about, using people. We don't have to worry about using a pancreas from another organism, which is how we used to harvest insulin. Okay, we used to use pig pancreases, and we'd have to use the insulin from that. Now we are able to absolutely safely, without the fear of things like contamination if it's done properly, bottle and make insulin, which is one of the things that genetic engineering is very useful for.